Hello everyone, let's get started. Today we're going to spend just a few minutes talking about colony morphology. Remember, we've defined this term colony to be a visible mass of microbial cells, and these cells are growing on a solid agar plate. All of the cells are clones of the parent cell. In other words, the parent cell has started to divide and produce daughter cells asexually until a whole pile or mass of genetically identical cells is sitting in this visible colony. We use solid media to grow bacteria in addition to other types of media, and depending upon the material that we use to inoculate that solid media, sometimes they will grow as colonies, and sometimes the bacteria will grow as what we call lawns. It all depends upon the starting concentration of the broth culture that we use to inoculate the agar plate. Take a look at this image right here on the slide. You can see two agar plates. The surface of both of these plates is not the nice sort of transparent beige color that we're used to seeing in an uninoculated agar plate. Instead, there's an opaque sort of yellow-white material growing all over the surface of this plate. This is what we would refer to as a lawn of bacteria. There are so many cells growing on the surface of this plate that rather than seeing these individual colonies, instead we just see this whole confluent mass of material growing. Sometimes we grow lawns because they can be useful to us in the laboratory. For example, if you look over at this plate is being used to test these bacteria for antibiotic sensitivity. We haven't talked about antibiotics yet, or how we test bacteria to see if they are susceptible to certain antibiotics. But this plate is showing a test being conducted for some kind of antibiotic that is within this little white disc. What they've done is they've created the lawn by smearing, again, culture material all over the surface of this dish. Very concentrated culture material, lots and lots and lots of cells in there. And then they took this little disc here that happens to be impregnated with an antibiotic. We can't tell what kind of antibiotic it is, but the disc is placed in the center of the plate, and then the whole plate got put into an incubator for approximately 24 hours. This is what the plate looked like the next day. What I want you to notice is that we did get a lawn of cells growing out here. Lots and lots and lots of cells crammed onto the surface of this plate, growing into this lawn overnight. But notice that there's a zone around that white disc where no cells have grown. This is an indication that these cells, these bacterial cells, are susceptible to this antibiotic, the antibiotic that has been placed into this little disc. Any cells that were present on the plate right around that disc at the time the plate was made have not survived. They have not been able to grow and divide. They died because the antibiotic killed them. It basically diffuses into the agar, and the cells, if they are susceptible to it, will not survive. Now you'll notice that these cells did survive. They're still susceptible to this antibiotic. It's just that they were far enough away from the antibiotic disc that they were able to survive and grow. So one of the big reasons that we make a lawn of bacteria on an agar plate is because we want to do some antibiotic sensitivity testing. We want to figure out if this particular microbe is sensitive to one or more antibiotics. But of course, other times when we make agar plates, we don't put nearly as many cells on. We put fewer cells because we want the cells to land far enough apart from each other on the surface of the agar that they're able to grow into individual colonies. Remember, we said we can create a spread plate for the purpose of counting colonies. That's going to help us determine how many microbial cells are in some volume of starting material. But we said we can also make what's called a quadrant streak plate just to grow some colonies. Not for the purpose of counting, but just to grow some individual colonies so that we can examine them for what we call colony morphology. Remember, that word morphology translates as body form. We can look at a colony and we can describe its physical, visible features. The features of a colony are one of the characteristics for that particular organism. Just like you and I have physical characteristics, like we have a certain height and we have a certain hair color, a certain skin color, a certain eye color. So too do colonies have certain physical characteristics. Typically, when we talk about colony morphology, we're talking about size and shape. We're talking about elevation, how far up above the surface of the agar the colony grows, and also about something called the margin or the edge of the colony, because that can take on different forms. On this slide, what you're looking at is a chart that's published in different source materials that shows different categories of colony morphology. When we examine a quadrant streak plate for colony morphology, we look at these categories, again, size, shape, elevation, margin, and then we use some of these terms to describe the differences that we can see from colony to colony. You'll notice that a lot of the terms on this particular chart are very strange looking to us. We see terms like rhizoid and umbonate and eros. There's all kinds of terminology associated with the morphology of colonies. And this is not the only chart that you'll find in textbooks and on websites regarding colony morphology. There's all kinds of source material to help us put scientific terminology on what a colony looks like. On this slide, you're looking at the surface of 4 different agar plates, and there are all kinds of colonies growing on these plates. There's a couple things to note here. This plate up here that has this short stretch of ruler on it, this plate, when you look at it, has several different visible types of colonies growing on it. In other words, the colonies don't all look the same. There's one kind of colony that's sort of large, and it has this whitish cream color. It's quite circular. The edge of it, or the margin of it, looks nice and straight. But nearby, there's a second type of colony, a much, much smaller colony. It's also white or cream-colored. It also has a nice even margin, but it's much, much smaller in diameter. And then there's a third colony on this plate. This one looks to have sort of a yellowish color, It's kind of halfway between the smallest and the largest colony in terms of its size. So this happens to be an agar plate that was made from an environmental sample. It makes sense that I can see multiple types of colonies growing on this plate because environmental samples usually have multiple kinds of microbes in them. When I'm evaluating a plate like this, I would want to note that I can see 3 distinctly different kinds of colonies on this plate, and I would note the features of each one. Again, the size, the shape, the margin, and the elevation. The important thing to remember, though, is that when I see 3 distinctly different types of colonies, I can only say that there are at least 3 different organisms making those colonies. And the reason I can only say at least is because it is possible for more than one species of bacteria to have the exact same colony morphology. In other words, these large white colonies, they may have been produced by one type of bacteria, but they may also have been produced by 2 different species of bacteria. I don't have any way of knowing that just by looking at the plate. Some features of colonies are quite common. We see them in multiple species. For example, the creamy sort of whitish color that you'll see is very common. The smooth edge around a colony, the smooth margin, is quite common. So just like it's very common for human beings to have, say, brown hair or brown eyes, we can see some features of colony morphology that are also very common from species to species to species. So I know that at least one species made this large white colony, but I can't say for sure whether multiple species have made that large white colony. So if I see 3 different, visibly different types of colonies on a plate, I can say there are at least 3 different species of bacteria growing on that plate. Now, I want you to take a look at this plate here on the right. I want you to notice something about these colonies. These colonies look kind of strange. And by strange, I mean I can see some colonies that are quite smooth around their edge, and others that have sort of an undulating edge, an uneven edge. I can see some that are small, some that are very small, and some that are quite big. In fact, it almost looks like no 2 colonies are the same. So how should I interpret this? Is each one of these colonies produced by a different organism? The answer is no. This particular group of colonies were all produced by the same organism. It's just that this particular organism, which is a bacterium, has the ability to secrete a very waxy material over the surface of its colonies. This is the organism that causes tuberculosis. So this is a kind of bacterium called Mycobacterium tuberculosis. And one of the features of this organism is that it secretes a waxy material. It's called mycolic acid, and it really does have the consistency of wax. And because it's secreting this wax, we end up with these very strange looking colonies, even though they are all produced by the same organism. Most of the time, though, when we grow colonies on an agar plate, in other words, for most bacterial organisms, all of the colonies that grow on the plate are going to be approximately the same size, approximately the same color, the same shape, the same margin, the same elevation, if they all belong to the same species. Down here on the lower left, I can see some colonies growing. They're all approximately the same size. They are approximately the same color, the same margin, and so on. This is probably one species of bacteria. In fact, it is one species of bacteria. This happens to be E. coli. I show you this picture for a couple of reasons. Number one, I want you to see that sometimes colonies end up growing sort of clustered together. And when you see colonies clustered like this and overlapping each other a little bit, it's really hard to determine colony morphology from this. Try to find individual colonies when you do colony morphology evaluation. Try to find 2 or 3 individual colonies and evaluate them. I say 2 or 3 because there will be some slight size differences between individual colonies. So if I measure 3 colonies in their diameter, and then I take the average, I'll have a good idea of what the size of these colonies is. I don't want to try to measure these over here because they're all kind of overlapping each other. It's much better to get these more individualized colonies to collect my data from. You'll notice they're all approximately the same color. They all appear to have a little bit of sheen on them. All of these things are characteristics of certain organisms. The other thing I want you to notice is that this agar is red. This happens to be what's called blood agar. And like the name suggests, this agar has had not only basic nutrients added to it, but it's also had blood added to it. Typically, that blood is sourced from sheep in the laboratory. So you can purchase sheep blood from chemical supply houses, and it can be added into the agar, giving it this red color. Remember, anytime we add an ingredient like blood or serum or milk or eggs or meat, we are creating an undefined medium. We don't exactly know all of the molecules that are present in blood. We just know that lots and lots of bacteria will use it as a food source. And the last thing to look at over here on this 4th plate is this beautiful red color in these colonies. This organism is called Serratia marcescens, and it has this wonderful characteristic of growing pigmented colonies. And as you can see, it's a very vivid red color. This is a characteristic of this organism, this red color. There are other organisms that produce pigments that turn the colonies colors, but Serratia is kind of distinct in its beauty. On this slide, you're looking at 3 agar plates. These were all taken, or I should say made, using environmental samples. And part of why I show them here, again, is to drive home this idea that most samples that are taken from the environment, whether we're monitoring a body of water or we're monitoring a hospital cleanliness program, most environmental samples are going to be mixed. In other words, there's going to be more than one type of microbe growing. So we have to be able to evaluate these dishes in terms of expecting to see multiple organisms. If you look at this plate, you can definitely see that there are different types of colonies here. They're not all the same. They're not all identical. Some of the things to notice, obviously, size. So I've got some colonies growing on here that are quite small. I've got others growing that are quite large. I've got different colors growing here. The edges of the colonies look all different. Some of them are smooth and even. Some of them are jagged. I've got some colonies that just look bizarre. They have really undulating margins, very strange shapes. Some of them have a central area that looks different from the rest of the colony. That's also a feature that we can see in some colonies. So I have to be prepared to, again, determine how many different types of colonies I see based on their features. And then report that I have at least that many organisms growing on the plate. Over here, I have another one, another dish, again, environmental sample, different types of colonies growing. I've got two colonies right here that have grown into each other. That's not unusual. Cells just landed here very close together. So as these two colonies formed, as the parent cells were dividing and dividing and dividing, they just sort of merged together. Sometimes you'll get one colony growing on top of another colony or sort of overlapping another colony. That happens as well. Generally speaking, the colonies that belong to a single organism are going to have essentially the same size, the same shape, the same color, so on and so on. So all of these colonies, this one, this one, this one, this one, and this one, and this one, here and here, these are all probably the same species, the same organism. It's a little harder to tell sometimes which of the other colonies are separate species, because they can all have very similar sizes, but if you look carefully, some of these are yellow and some of them are white. So these are probably two different organisms. If you take a look at this plate right here, this is also an environmental sample. Here, I'm going to move this one out of the way just for a second. A couple things to notice. Number one, what a well-labeled agar plate. You can see that there's lots of information written on this plate. You remember, you always want to put your name. You always want to write down what kind of media the agar is, what type of agar you've got in the dish. You also want to write down what's growing. And in an environmental sample, of course, you're not going to know what's growing until you evaluate it, but you can write down where it came from. So this was a swab plate that was created by sampling a dust pan. And the swab was then smeared over the surface of the agar to grow some colonies. There's a tremendous variety of size and shape on this plate. There's some kind of organism growing over here that doesn't really look like a colony. It's all just sort of merged and growing over this large area. This is a reminder to us that bacteria are not the only organisms that can grow on a nutrient agar plate. The fungi can also grow on these plates. And while we think we could tell the difference between bacteria and fungi, we really can't. You really can't just by looking at the colonies. You need to take a sample from the colony, make a smear, stain it, and examine it under the microscope. I want you to look at this strange colony right here. This is one colony, but how unusual. It doesn't look like a nice circle. It doesn't have an even edge. It's quite unusual. It doesn't have any kind of a symmetric look to it. And there's more than one of these on this plate. There's one here. There's another one here. There's one here and here. There are many different organisms out in the world. And while we sample from the environment, we have to expect to get some unusual sizes and shapes, unusual colony morphology. Over here we have another agar plate. I show you this one because it shows an attempt to determine how many different colonies are growing here. And someone has overlaid this plate with numbers to show each individual type of colony that they can see they've identified this sort of mid-sized yellow colony as number 1, and then there's another mid-sized, more white colony labeled number 2, a very small colony 3, over here at 4, something larger, all the way up 5, 6, 7, 8, 9, 10, 11, 12. This tiny little white colony, there's lots and lots of them growing here, is number 12. So this can be hard work. It can be hard work counting up the different kinds of colonies on an agar plate, and it takes some experience. Look at this unusual looking colony, number 8. Very strange looking, a strange color. It's got an area in the center that looks different from the rest of the colony. Very unusual looking. So, very important for us to remember that just by looking at colonies growing on an agar plate, we can't say whether they're fungi or bacteria. If you look at this picture, for example, I could see 2 colonies growing on this blood agar plate. And boy, if I had to guess, I would guess that these are fungal. They look kind of fuzzy. They're not particularly symmetrical. They have that whitish sort of powdery look to them. Boy, I'll bet these are fungi. And if I look over here on the left-hand side, I can see an agar plate with, again, lots of different kinds of colonies growing on it. And boy, I would guess that these are bacterial. They're nice and symmetrical. A lot of them are round. Lots of different colors, but really you might look at these and think, oh, these are bacteria for sure. In actuality, this plate is full of fungal colonies, and this plate has a bacterium growing on it. So again, you just can't tell bacteria from fungi by colony morphology. You have to do further testing. You have to look at the organism under the microscope to know what you're dealing with. We're not going to spend a lot of time in this course practicing colony morphology. I just want you to be aware of what it is and how it's done and the way we interpret it, especially the idea that if we see 3 or 4 distinct kinds of colonies on a plate, we never make the claim that that means there are 3 or 4 organisms on that plate. We always hedge that by saying at a minimum, there are 3 or 4 organisms growing here. We always recognize that multiple species can have identical colony morphology.